maybe the second best meeting in breast cancer. I don't know what the first one might be, but it's a close tie. Yeah, so. Okay, so it probably would have been better for my talk to follow Jose's because he gave part of my talk in a sense. I thought he was going to talk more about HER2-positive disease, but we're going to approach this from a different manner, so maybe there is room for both talks. And what I'm going to try and convince you is that PTEN loss is not only important in triple-negative breast cancer, where we thought that it's important, but it's also important, I think, in ER-positive breast cancer, not due to mutation, not due to complete loss, but due to slight downregulation that can really activate the PI3 kinase pathway, much like a receptor tyrosine kinase overexpression can. Okay, so the way I think about treatment resistance is that in cancer, there's likely to be a driver pathway, and this is a molecular pathway or network that is the main stimulator of proliferation and survival of the cancer cell, i.e. estrogen receptor and HER2. And there's also escape pathways. These are pathways that function along with the main driver to stimulate tumor growth, or that take over and become more active when the driver pathway is blocked. Now, in the case of a cell that's estrogen receptor positive, there are multiple escape pathways that could be functioning to contribute to less responsiveness or resistance to various endocrine therapies. And first, obviously, we have the nuclear ER signal coming in here to activate ER, and it binds to EREs, transcription elements, on target genes to stimulate transcription and cause tumor growth. And this is the classical ER pathway. Well, estrogen can also activate ER outside the nucleus, and that can activate receptor tyrosine kinase pathways like the PI3 kinase pathway. And when you add estrogen to cultured cells, with just a few minutes, you activate the PI3 kinase pathway. And there are a whole variety of other pathways that function in a cell to keep it alive when an important pathway is blocked: the receptor tyrosine kinase pathways, PI3 kinase AKT, or RAS, MAP kinase, signals coming from the microenvironment, integrins, stress pathways. And they can all induce their own genes by induction of transcription factors that can contribute to proliferation and survival of the cancer cell. But in terms of ER-positive breast cancer, they also have other very important functions that can contribute to resistance. And that is, they phosphorylate the estrogen receptor and its co-activators, such as SRC3. SRC3, which is probably just as important as ER, and we're learning how to target it now for treatment purposes, has 11 phosphorylation sites. ER itself has 8 or 9. And if you think about it, that means there's 8 or 9 different estrogen receptors, and there's 10 or 11 different SRC3s, depending on what sites are phosphorylated to activate transcription. And there are several things that happen when you activate the estrogen receptor by some of these pathways. And those are summarized here. First, when you activate the PI3 kinase pathway, ER and PR levels decline. And they decline because AKT phosphorylates FOXO-proteins, and then FOXO-proteins come out of the nucleus. FOXO-proteins are very important for expression of the ER gene. So it's logical that when you activate AKT, ER levels are going to drop. Secondly, ER is activated by phosphorylation by many different factors that I just mentioned in a ligand-independent way. Third, ER moves from its classical EREs on DNA to other sites, such as FOXO-A1 or AP1, to activate a totally new transcriptional program. And we published, 4 or 5 years ago, that you look at the gene signature induced when cells become resistant to tamoxifen. It looks for all in the world like a HER2 amplified gene signature with a variety of growth factor genes being turned on by the estrogen receptor. And then finally, as Jose pointed out and others point out, tumors like that have activated PI3 kinase pathway and ligand-independent activation of ER are still dependent on ER. When you siRNA ER, the cell dies. It's very effective therapy, even though none of our endocrine therapies work. And so tamoxifen or aromatase inhibitor is not going to work in that setting. They'll be ineffective. But drugs that are selective estrogen receptor downregulators if we can really get a good one that can be given at a sufficient dose to really downregulate ER, I think, are going to turn out to be the most important therapy. Now let's talk a little bit about the PI3 kinase pathway, which is shown here. And this pathway can be activated, as you know, by upregulation of receptor tyrosine kinases, by downregulation of PTEN, maybe by activating mutations in PI3 kinase or AKT. And activation of this pathway initiates a phosphorylation cascade that leads to enhanced protein synthesis and cell survival. But what also is important to recognize here is that there are negative feedback loops. All biological systems have rheostats that can turn them up or turn them down. And there are negative regulations here, negative feedback loops. One going from S6 kinase to mTOR, mTORC2, which can activate AKT. It's an upstream of AKT. There's another one from S6 kinase that can block IRS1 and activation of the PI3 kinase pathway. And there's another one that's been described recently that can downregulate the expression of receptor tyrosine kinases. So this is a very complex pathway. And I think, as you'll see in a second, the point I'm trying to make is that PI3 kinase activating mutations, shown here, are probably by themselves weak activators of the pathway. And these negative feedback loops then become function and essentially turn off the pathway. So if you think in ER-positive breast cancer, I'm beginning to think that a PI3 kinase mutation is an inactivating mutation if you consider the entire pathway in ER-positive disease. Now, it's got to be important, right? Because it was selected during the process of cancer to be expressed in certain cancers. So it's got to be important. And maybe, as Jose pointed out, it functions better in collaboration with other activators of the pathway, like HER2 amplification, as the example that he showed. So the literature on this is very confusing. You've got to read every paper about 10 times before you sort of sort things out. But this is my take on the role of PI3 kinase or PIK3CA mutations in breast cancer. It's very common, as you've heard, but it's more common in luminal A tumors that we all know have a better prognosis. And it's associated with a high ER and PR, negative HER2, low proliferation, and a better prognosis and good response to endocrine therapy. So what does this mean? What does a PI3 kinase mutation mean in these tumors? They have a better prognosis in most of the large, recently done trials. Here's a study published by Loy and colleagues that demonstrated this. On this side, this panel are a graph of all tumors that they looked at with PI3 kinase mutation or wild type. But when you focus in on the ER-positive breast cancer subtypes, and you divide these wild type tumors into low proliferation or high proliferation, you get this kind of a thing. These are the low proliferating wild type tumors. Here's the high proliferating tumors. And here are where all the tumors with PI3 kinase mutations fall. They're more like low proliferating wild type tumors. They have a good prognosis. So if that's true, then what else can activate this pathway that would be bad for endocrine therapies? Well, we think PTEN can do that. And we're hypothesizing that PTEN, which is, I think, a stronger activator of the PI3 kinase pathway than PI3 kinase mutations are, that PTEN expression levels may better dictate the sensitivity to endocrine therapy in ER-positive, HER2-negative tumors. So it raises some questions. Does PIK3CA mutation status differentially correlate with PTEN expression levels? And is complete loss of PTEN required to activate the PI3 kinase pathway? The answer to both of those questions is no. This is a paper that Gordon Mills's group published several years ago that I think demonstrates a lot of important findings. And it shows here a set of tumors with mutations in PIK3CA or different levels of PTEN going from low to high, and evidence of activation of the PI3 kinase AKT pathway by level of phospho-AKT. And this, I think, is interesting because it demonstrates a variety of features. And that is, if you have a PIK3CA mutation, it can fit anywhere on this spectrum. You have PIK3CA mutations that are associated with high expression of PTEN or low expression, and with either low levels of phospho-AKT or high levels of phospho-AKT. There seems to be no relationship. On the other hand, P10 levels do relate to this, and low P10 levels are associated, as you might guess, with high expression of phospho-AKT and activation of the pathway. But you can note that activation of the pathway is even way down here at P10 levels that are not particularly low. And in fact, in experimental models, a 30% reduction in the messenger RNA for P10 can activate, and messenger RNA for P10 really correlates well with protein, can activate the PI3 kinase pathway. This is a study that we published several years ago, along with Gordon Mills, where we developed a protein signature and a transcription signature reflecting a PI3 kinase score. And the higher the score, you can see the yellow here, as evidenced by various phospho-AKTs, phospho-mTOR, et cetera, or by genes that are either repressed or induced by PI3 kinase inhibitors, that increasing score of the PI3 kinase pathway, activity of the pathway, is associated with ER loss, which we had shown before this, with HER2 expression causing upregulation of the pathway. And as you have a higher PI3 kinase score, the tumors become more and more luminal B-like. And that's true with both the protein signature and the RNA signature. That activation of the PI3 kinase pathway is more common in luminal B and is associated with downregulation of ER and PR, as you might expect. Loy's paper showed a similar thing. Here are luminal A tumors having a high level of a PIK3CA mutation gene signature, whereas luminal B have a low. And remember, this signature is one that turns off the pathway. And here's a P10 loss gene signature, which is more common in luminal Bs than it is in luminal As. And then finally, a recent paper presented by David Rim at the San Antonio meeting this year, looking at quantitative levels of P10, either by RNA or by protein. And he shows the same thing. That is, if you have low levels of P10, which activate the PI3 kinase pathway, you have a worse prognosis. And that's true with the RNA, maybe less true with protein, but still true. And the cutoff for the protein was the median. It wasn't no expression or very low expression. It was the median of P10 expression. Obviously, this needs to be worked out in a little more detail if you could have a clinically useful assay. But this suggests that low levels of P10 but not complete loss are associated with a worse prognosis, probably because they activate the PI3 kinase pathway. So to study this a little bit more, we developed a P10 knockdown model of ER-positive breast cancer where we could establish P10-inducible knockdown cell models, look at the endocrine sensitivity of these in culture, look at the endocrine sensitivity in xenografts, explore various inhibitors or alone in combination, and the inhibitors we chose were an mTOR inhibitor, an AKT inhibitor, and a MEK inhibitor that were supplied by AstraZeneca. And we looked at kinase inhibitors combined with endocrine therapy, and then in vivo validation of what we thought would be the most potent regimen. And here's a series of cell lines. It's interesting that you almost have to have a PI3 kinase mutation or P10 loss to be adapted to tissue culture. So 20% of breast cancer cell lines have P10 mutations and loss. These cell lines don't, but they have PIK3CA mutations, and this cell line doesn't have either. And what we're doing is using these cell lines to knock down P10 to see what the effect is on endocrine sensitivity, and then to look at what drugs that block various components of these pathways, including the feedback loops, might be optimal for a given endocrine therapy. You can see that these cell lines that have P10 loss do indeed have upregulation of the PI3 kinase AKT pathway with upregulation of phospho-FOXO3A, phospho-AKT, et cetera, and they tend to have lower levels of estrogen and progesterone receptor. So this is the construct we use to use an shRNA for P10 to knock it down and activate the PI3 kinase pathway. And if you look, this just summarizes the data to this level. They do have lower ER and PR when you knock down P10. Interestingly, they have a reduction in total amounts of AKT, but they have increased phospho-AKT and activation, other evidence of activation of the pathway. And they have activation of the MAP kinase pathway in some lines. When you reduce P10, you activate not only the PI3 kinase pathway, but the MAP kinase pathway in some cell lines in tumors. And interestingly, they also have increased phosphorylation of c-Jun, which is important in the AP1 transcription factor complex. Well, this shows several of the cell lines and increasing doses of doxycycline to reduce PTEN levels and activate the pathway. And what this shows is that even with relatively low doses of doxycycline and relatively modest reductions in PTEN, you have evidence of activating the PI3 kinase pathway with phospho-AKT in both places there, FOXO3A, GSK3B, and even MAP kinase in this cell line. And the same with the MCF7 shRNA for PTEN. You can activate, you reduce ER levels, you activate phospho-AKT, FOXO proteins, and an increase in MAP kinase, although with very high doses of the doxycycline to really reduce PTEN levels, you actually have a reduction in phospho-MAP kinase. So this is the model, and what does it do for endocrine therapy? Well, first of all, if you're knocking down PTEN and activating the pathway as shown in the red, and it has no effect, doing that has no effect on estrogen-induced growth. I mean, estrogen itself activates the pathway, so further activation of the pathway doesn't seem to matter. But when you take away estrogen deprivation, now you can see with estrogen deprivation or tamoxifen or with fulvestrant, the cells in red are less sensitive to inhibition by the endocrine therapy. Fulvestrant, you'll notice, seems to be the least affected here, probably because it is active in inhibiting the ligand-independent activated estrogen receptor. In a clonally formation assay, similarly, in red is knocking down PTEN, and with estrogen deprivation works well without it, but when you knock down PTEN, it causes relative resistance, except with fulvestrant, where, yes, it does make fulvestrant somewhat less effective, but fulvestrant in these doses is the most effective endocrine therapy. What about in vivo? This panel up here is those without PTEN knockdown. You can see that estrogen deprivation in vivo is very effective. One tumor escaped over a year. Tamoxifen is a little less effective in this model. It inhibits growth for about 2 to 3 months, and then tumors escape. And fulvestrant is very effective. And then when you knock down PTEN with doxycycline, you can see that these endocrine therapies are much less effective with tumor escape very quickly, fulvestrant being the most effective agent. So that led to some questions. Do various inhibitors of the pathway work better if the pathway is hyperactive? Which inhibitors work best? Given the many feedback loops, are combinations of inhibitors required? And given the interaction with ER, what is the best endocrine therapy to use when this pathway is activated? And just a few, I just don't have time to show you very many experiments, but here's one. This is estrogen treated in the absence of knockdown PTEN, and these drugs are very effective. Of course, these cell lines all have PI3 kinase mutations, maybe that's the reason, but an mTOR inhibitor works pretty well, AKT inhibitor less well, a MEK inhibitor less well, and combinations of mTOR and AKT work particularly well. When you then turn on the PI3 kinase pathway, these cells are not more sensitive to these agents, they're less sensitive to these agents, as shown here. Here's the mTOR inhibitor when you activate the pathway compared to here. And you can see the same with the other drugs. The most effective combination between being the mTOR and AKT inhibitor. And now, what about estrogen deprivation? Adding in an endocrine therapy, well, it's very effective itself, but it's more effective with an mTOR inhibitor. That would be akin to using in the BOLERO-2 trial everolimus along with estrogen deprivation. AKT inhibitors, it helps a little bit. MEK inhibitors less so. The combination of mTOR plus AKT is the best. And when you turn on the PI3 kinase pathway, again, with estrogen deprivation, it's less effective, but they're still relatively effective. Here's the mTOR inhibitor. And again, the most potent combination is mTOR and AKT, shown here. And then what about tamoxifen and fulvestrant? They're shown here. This is in the absence of activation of the PI3 kinase pathway. In this model, tamoxifen is pretty effective. Adding in the pathway inhibitors make it somewhat more effective, particularly the AKT and MEK inhibitor. And for some reason, it depends on which endocrine therapy you use. And with fulvestrant, it's the same way. AKT and a MEK inhibitor is the most important. AKT inhibitor by itself is pretty effective. When you upregulate the PI3 kinase pathway, a similar thing. With tamoxifen, the AKT plus MEK inhibitor is best, and with fulvestrant, either the AKT or the AKT plus MEK inhibitor is best. So you can play around with these drugs in various ways, and we've done a whole series of experiments that I don't have time to tell you about, where you look at different doses of all the agents used in various combinations, and then you can come up with what are the most synergistic compounds. And that may give some clue as to what we should try in the clinic. You saw that list of drugs that Jose showed. How are we going to ever study all these drugs? Well, preclinical models are not perfect, but they may help provide clues as to which drugs we can choose and hypothesize would work better in people. This is an in vivo study with what we think is the most effective inhibitor, and these are SHP10 knockdown MCF7 cells, looking at the most effective combination, fulvestrant plus an AKT inhibitor. Adding in a MEK inhibitor makes it a little bit better. But notice this curve is not going like this, like you saw in many of the slides before. This curve is going down, and we need to carry it out for a longer period of time. But that suggests that we actually are inducing apoptosis with the combination of the fulvestrant plus the AKT inhibitor. So to conclude, we think that reduction in PTEN, but not PI3 kinase mutations alone, activates the PI3 kinase AKT signaling pathway, reduces ER in classic ER-regulated genes. Complete loss of PTEN is not necessary. A clinically useful assay for PTEN loss or PI3 kinase pathway activation, I think, needs to be developed and would be very helpful. Activation of the PI3 kinase AKT pathway leads to relative resistance to a variety of endocrine therapies in vitro and in vivo. AKT, mTOR, and MEK inhibitors enhance endocrine therapy, but this is attenuated to some extent by PTEN loss. So tumors that have high activation of this pathway are going to be less sensitive to these pathway inhibitors, not more sensitive. So at least this data would suggest that. The optimal inhibitor combination depends on type of endocrine therapy and PTEN level. And finally, fulvestrant plus an AKT inhibitor or plus adding in a MEK inhibitor is the most important, most potent combination in vitro and in vivo, whether or not the PI3 kinase AKT pathway is activated, and I think deserves clinical trial. And in fact, the trial that Jose showed at the end of his talk, I think is a great trial. Our data would suggest that would be, I think you're using a PI3 kinase inhibitor, but a AKT inhibitor would probably show the same thing. So I'll stop there and thank the people in my group who contributed to this work.